Good evening, people. How are you? Can you hear me clearly? Hello, hello, living beings. How are you, people? Komuda. Can you hear me, all of you? Is it clearly audible? Abdullah, Amira, thing. So, how was that the lesson today, people? Coordination. You think you can? Yeah, you can get this the lesson. Little different lesson, eh? Little different lesson. Um, so people, Kaumuditi. So, right. So, people, today we're going to discuss paper number 13. Did you answer the paper? Did you answer the paper? I send you the marking scheme. I'll give you the printed one on Saturday. There was a small delay in printing the marking scheme, but I'll give you this marking scheme so you don't have to print that. I'll give you the printed one on Saturday. Did you answer the paper? Karada, Yutika, did you do this paper? Karada? Yes or no? Yes, Yutika Karalu. Anyone else to do the paper 13? Huh? Anyone else to do paper 13? Anyone else to do this paper 13? Just give me 1 minute, people. I didn't bring my pens up. Just give me 1 minute. 1 minute, please. Answer. Answer. Anything? Hurry, hurry. Sorry about that. Still the pens in my car. Couldn't bring it here to the classroom. And just came home, people. How about you? You went 2 p.m. How about you, Mr. Rimas? I just came home. I had another class in the afternoon, small class, 27 students, weekday class. Um, so let's go to the board, ne? Let's go to the board. Let's go to the board. Right. So people, paper number 13. Paper number 13. Paper 13. Let's see. Right, people. Right. Paper number 13. Shall we check that? Paper number 13. Let's check that. Right. This is the paper. Paper number 13. Yes, check your answers. Right. First question. First question. Which of the following compound is the monomer? Which of the following compound, the monomer is a ketose? Which of the following compound, the monomer is a ketose? Glycogen, hemicellulose, chitin, inulin, and pectin. What is the answer, people? What's the answer? Which of the following compound monomer is a ketose? Now you learn two ketoses. You learn two ketoses. Who's going to type me the two types of ketoses that you know? One is in the resource book, other one is not in the resource book. Can you tell me those two ketoses? Who's going to tell? Who's going to tell? No, you have to, now you have to use the main chat. Kavya. Good. Kavya, good. Can you use the main chat? Amira, can you come to the main chat? That's like easier for me. Otherwise, I have to like click your answer. So one is fructose. Kavya says fructose. Kavya says fructose. Kavya says fructose. Mr. Rimas also says fructose. Do you know anything else? Do you know anything else? Um, any other ketos? Do you know any other ketos? Ribulose, very good. Devmi says ribulose. Umara also says ribulose, very good. Right, ribulose. So here we can take then the answer as the glycogen. Glycogen is a glucose polymer, so you can't take that. Hemicellulose you can't take. It's a pentose sugar polymer. Chitin, glucosamine. Inulin is a fructose polymer. Inulin is fructose polymer. So we can take inulin. Inulin as the answer. Right. Moving to question number 2. Which of the following statement regarding light microscope is correct? Light microscope, correct statement. Light microscope. Which of the following microscope correct? Cannot be observed by which of the following statement regarding light microscope is correct? Cannot be observed by naked eye? No, you can neither. Actual colors of the object cannot be observed. Correct, Nahani? Cannot be observed by naked eye. Wrong. Actual colors of the object cannot be observed. Actual colors you can see, neither. You can see actual colors. Less resolution power than the electron microscope. That is correct. Resolution is lower. Remember the resolution power. Can someone tell me the resolution power of the light microscope? Who's going to tell me the resolution power of the light microscope? Valib, I won't. I need the valib. Anyone to tell the resolution of light microscope? Huh? What's the valib? Valib. Electron microscope. What we said for electron microscope, how many nanometers? 2 nanometers, right? Electron microscope, 2 nanometers. So how about the light microscope? 0.2 micrometers. That is 200. That is 200 nanometers. 200 nanometers, right, people? So less resolution power than electron microscope. That is correct. Heavy metals are used for staining. That is wrong, neither. We don't use heavy metals for staining. And then that's we are using for electron microscope. And then strong magnets are used. That is also wrong. Strong magnets are used. That is also wrong. So we have the answer now. The question number 2 answer is third one. Question number 3. Select the correct statement about eukaryotic nuclear division. Eukaryotic nuclear division, correct statement. Nuclear envelopy is fragmented and centromeres are located in the metaphase plate during pro-metaphase. Nuclear envelope is fragmented. Nuclear envelope is fragmented. They are asking correct statement. Correct statement. Correct statement. Nuclear envelope is fragmented and the centromeres are located in the metaphase plate during pro-metaphase. That is wrong. We can't agree with that. Right? Nuclear envelope fragmentation occur during the pro-metaphase is correct. But then the centromeres are present in the metaphase plate during pro-metaphase. We can't agree with that. Next one, we will kinetoko. Kinetoko microtubules move chromosome back and forth during anaphase. That is wrong, neither. Where do you see that this back and forth thing happens? Back and forth thing happens during the pro-metaphase. So therefore, that is also wrong. That is also wrong. Right? So we'll take this kinetoko microtubules move back and forth wrong. Both chromatids of one chromosome are attached to the kinetoko microtubules from one pole in the prophase 1. Both chromatids of one chromosome are attached to kinetoko microtubules. Both chromatids of one chromosome are attached to kinetoko microtubules from both poles. That is correct. For mitosis, this is correct. Meiosis, this is not correct. Mitosis, this is correct. So both chromosomes of one chromosome are attached. We'll take it as correct. Sister chromatids of one chromosome move to the same pole in mitosis anaphase. Sister chromatids of one chromosome move to same pole during mitosis. Sister chromatids of one chromosome move to same pole. Wrong, neither. That doesn't happen in mitosis. Maybe you can take it as in meiosis. Wrong statement, people. Can anyone resend the scheme? I said it. I said it. Okay, I'll send resend. Okay, got it. For the Abdullah. And so next one, sister chromatids of one chromosome move. Sister chromatids of one chromosome move. Wait, people, because when I mark, what happens? When I mark, right now, okay. Now, okay. That's little pros on it. Okay. Let me move. Sister chromatids of one chromosome move the same pole. Wrong. Genetically non-identical. Genetically non-identical 4 daughter chromosomes. Nuclei, sorry. Genetically non-identical 4 daughter nuclei are formed at the end of the anapase 2. Genetically non-identical 4 daughter nuclei are formed at the end of the anapase 2. That doesn't happen in mitosis. So what is this question, people? Select the correct statement about eukaryotic nuclear division. They just asked for nuclear division. So sister company, this one will take it as wrong. And then genetically non-identical 4 daughter nuclei are formed at the end of anaphase 2. End of anaphase 2. End of anaphase 2. Oh, people, end of anaphase 2. End of anaphase 2, you can't say that the nuclei are formed. End of telophase, we can say that the genetically non-identical 4 daughter nuclei are formed if you consider mitosis. Sorry, if you consider meiosis, right? So therefore, I'll take this statement also wrong, right? This statement also wrong because not at the anaphase 2, telophase 2. Because the question asks regarding generally about the nuclear division, not mitosis or not meiosis. Is that okay? Right. So my answer is third one. My answer, I'm taking people third one. Answer is third. Right. Question number 4. Question number 4. Which of the following correct regarding allosteric regulation of enzymes? Allosteric regulation of enzymes. Right. Check them, people. Which of the following correct regarding allosteric regulation? These are made up of one or more subunits. One or more subunits, wrong, neither. They're asking correct statement. Can't be one, neither. 2 or more subunits. So I'm taking it as wrong. And then, people, the regulatory molecules affect only the shape of the enzyme. Regulatory molecules affect only the shape. Function also, neither. And then activity can be activated or inhibited by binding one substrate molecule to an enzyme. The activity can be activated or inhibited by binding one substrate molecule to an enzyme. What do you say about that? What do you say about that? Right? The activity can be activated or inhibited. Activated or inhibited by one substrate molecule. Now, we know in allosteric activation and inhibition, they don't go and bind with the active site. They don't go and bind with the active site. They bind with the regulatory site, allosteric site. Right? They bind with the allosteric site. Activity can be activated or inhibited by binding of one substrate molecule to an enzyme. Doubtfully, we'll just go with the other statement. Regulatory molecules bind to specific regulatory sites of an enzyme irreversibly wrong. Irreversibly wrong. We can't agree with that. And then intermediate produced in metabolism involved in regulation of the production of more end products than required. Intermediate products of metabolism involved in regulation of production of more end products. Involves in regulation of production of more end product. Not intermediate. What do you say? End product. End product. So we don't have any other answer. The best answer, people, is the third one. Best answer is third one. True that this third one is not also the perfect one, but that is the closest answer we can take, right? The activity can be activated or inhibited by one substrate molecule. But if you check the resource book, I think for cooperativity, maybe we can agree with this substrate molecule bind with active site. Cooperativity, when you check that, people, in your resource book, cooperativity. Shall we check the cooperativity? Enzyme action cooperativity. Let me read that for you. Let me read it, Lamai. Cooperativity. It says for cooperativity, this is another type of allosteric activation. Binding of one substrate molecule can stimulate binding or activity at the other active site. But mitana beta tino neither activated or inhibited story inhibited by binding of one substrate. Inhibited is not mentioned in the resource book, right? Inhibited is not mentioned in the resource book, right? Stimulated binding or activity at other binding or activity inhibited issue. So anyway, we don't have any other alternative answer here. So I'll take the third one as the best answer for this question, but with some issues, right? With an issue, right? Do you have any other alternative answers, best answers, any suggestions, people? What did you mark? What did you mark here for fourth question? Any other alternative answers that you have marked? Yes or no, people? Right. No, sorry. Shall we go to fifth question then, people? Fifth question. Fifth question. Which one of the following statement is correct regarding anaerobic respiration? Correct regarding anaerobic respiration. You know anaerobic respiration, fermentation, neither. Anaerobic respiration is fermentation. In absence of molecular oxygen, anaerobic respiration take place, right? So ethyl alcohol fermentation and lactic acid fermentation. Fermentation means final hydrogen and the electron acceptor is an organic compound. So you call it as a fermentation. So here, which of the following statement is correct regarding? Which of the following statement is correct regarding? Which of the following statement is correct regarding anaerobic respiration? Anaerobic respiration. In the ethyl alcohol fermentation, primary hydrogen acceptor is pyruvate. Ethyl alcohol fermentation, primary hydrogen acceptor is pyruvate. Ethyl alcohol fermentation, hydrogen acceptor is acetaldehyde, so it's strong, right? Hydrogen acceptor is pyruvate, wrong. End products of lactic acid fermentation are lactic acid. End product of lactic acid fermentation are lactic acid, ADP and NADPH, wrong. We can't agree with that also. End products of alcohol fermentation are carbon dioxide, ATP and NADPH. End product of alcohol fermentation are carbon dioxide, ATP and NADH. Carbon dioxide, ATP, NADH, people also we can't agree with that. Alcohol fermentation are carbon dioxide, ATP and NADH. Alcohol fermentation, carbon dioxide. ATP is synthesized when pyruvate is converted to lactic acid. ATP is synthesized when pyruvate converted to lactic acid. when pyruvate converted to lactic acid. Wrong, neither. That doesn't happen either. And then hydrogen released from one molecule of glucose. Hydrogen released from one molecule of glucose reduce hydrogens released from one molecule of glucose reduce 2 NAD into 2 NADH. Hydrogen. How many hydrogen actually released here? The resource book, what it says, people? 4 hydrogen release, ne? 4 hydrogen release and 2 NADH is going to form. 4 hydrogen release and electrons release and then form 2 NADH molecule. So we can agree with the fifth one. So fifth one is the answer. Fifth one we can take as the answer. Right? Fifth one is the answer. Shall we move to next page, people? Let's move to next page. Something wrong with this setup today. Listen. Right. It stuck a little bit. I don't know the reason. Right. We'll move to next page, people. Next page, question number 6. Question number 6. The classification of Robert H. Whittaker. Classification done by Robert H. Whittaker. Kingdom Protista was introduced. Protista. Whittaker's 5 kingdom classification. Ne? 5 kingdom classification. What are those 5 kingdoms? The Monera, Protista, Plantae, Animalia, Fungi. Ne? So the classification of Robert H. Whittaker. Kingdom Protista was introduced. Kingdom Protista was introduced. And then Kingdom Protista was introduced. How about that? That is wrong, neither. That is wrong. Kingdom Protista was introduced. Then domain bacteria was introduced. Domain bacteria wrong in the Carl Bose classification. Kingdom Monera and fungi was introduced. Kingdom Monera and fungi. How about that, people? Monera and fungi, that is okay. Ne? Microorganism was first classified. Wrong, neither. Whose classification they talk about? Ernest Haeckel, ne? With the discovery of the microscope, Ernest Haeckel. So that's wrong. Theories of evolution were considered. Whittaker's classification, we said that with the establishment of the evolution theory, from Carl Bose classification onwards, we can see that this evolution theory was used in classification. So this is what the resource says about this classification. Classification history. It says with Ibn statement Taklamai. With the acceptance of Darwin's theory of evolution and unitary origin of life, taxonomy began used to natural system, which interpretation of evolution relationship with the recent advancement of molecular biology and molecular methods in studying. So then Carl Bose one, 1977, Carl Bose. So that is not describing people. We take as classification. So we have the answer. We can take third one as the answer. Third one as the answer. Right? Question number 7. Question number 7. Question 7. Question 7. Which of the following is not a common characteristic of organisms belongs to both domain archaea and eukarya? Not a common characteristics of organism belongs to both domain archaea and eukarya. So check these statements, people. Check these statements. Check this. Growth is not initiated when antibiotic streptomycin is present. Now you are comparing archaea and eukarya. Now in archaea and eukarya, both we know people growth is not inhibited by antibiotic streptomycin. So it's a common feature. Ne? So you can't take it as not a common feature. This is wrong. Not neither. They say not a common feature. Then initiate amino acid of protein synthesis is methionine for both. So you can't take that either. Several kind of RNA polymerases present. Several kind of RNA polymerase is a common feature. So you can't take that. And growth occur above 100 degrees Celsius. This is not a common feature. Presence of cell walls made up of polysaccharide. Presence of cell wall made up of polysaccharide. How about that? How about that? Right? Presence of cell walls made up of polysaccharide. What we said in archaea, different proteins and polysaccharides. Archaea cell wall material, people, it says. Archaea cell wall material. Cell wall material, proteins and polysaccharides. Eukarya cell wall materials, hemicellulose, cellulose, and pectin. So hemicellulose is a polysaccharide, so you can't take that also. That is presence of cell wall made up of polysaccharide, common feature. So you can't take that as the answer. So we got the answer here. Fourth one as the answer. Fourth is the answer. And moving to question number 8. Question number 8. Question 8. Which each of the following can be seen in some groups of kingdom Plantae? Can be seen in some groups of kingdom Plantae. When each of the above features are arranged as seen in 1, 2, 3, or 4 groups of kingdom Plantae, the correct sequence is each of the following feature can be seen in some of the groups, right? Each of the following feature can be seen in groups of kingdom Plantae. Heterosporous. Now, what are the phyla with heterosporous condition? Right? How many phyla are there people with heterosporous condition? Let's take the order name. If you take Bryophyta, homosporous, you can't take that. And then if you take people, if you take Pterophyta, homosporous, you can't take. Lycophyta, both neither. Homosporous one members are there, heterosporous ones are present. Can be seen in some of the groups of kingdom Plantae. Which are the following seen in 1, 2, 3, 4 kilahane? 1, 2, 3, and 4. So heterosporous you can see present in some Lycophyta, Psychodophyta, Coniferophyta, Anthophyta. Heterosporous people, I get number 4 here. Heterosporous, I get 4. Process of xylem vessel element. Xylem vessel element is only available in 2. I forgot to mention Nettophyta. Nettophyta also there. So we'll try that once again. Heterosporous condition. Heterosporous, Lycophyta, Psychodophyta, Coniferophyta, Nettophyta, Anthophyta. So I get now 5 here. But little doubtful to take whether Lycophyta or not, but I'll just write 5. Presence of xylem vessel elements. Presence of xylem vessel elements. Xylem vessel elements. Vessel elements are present in Nettophyta. And what is the other one, people? Nettophyta. Vessel elements in Nettophyta, and what is the other one? Right? Nettophyta, what is the other one? Come on. Come on. What's the other one? Nettophyta and Anthophyta, right? Xylem vessel element. Vessel element. Vessel elements are present in Anthophyta and in Nettophyta. Vessel elements, so I'm taking process of xylem vessel elements, I'll take it as 2. Then dominant gametophyte. Bryophyta has, there is a question, people. Groups, groups. Not phyla, Now there is a problem. Let me show you that issue. Let me show you the issue. Groups. Open with. Right. Let me show you the issue, right? This is, they say groups, ne? Groups. Let's see plant groups according to resource book. Let's check the plant groups. According to resource book, plant groups. According to resource book, plant groups. What do you think? Right? Now they don't say phyla, ne? If they say phyla, people, no issue. But that's not phyla, ne? I think they based on this. Oh, this one. Maybe in the plant groups. Oh, 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 plant groups. Ah, here based on this plant groups. Let's see now. Let's check the question once again, people. I'll keep it like this. Plant groups. Now, one of the group is non-vascular. Bryophyta, Pogonatum, Menthoceras, and Markentia under that. Another vascular plant. Seedless vascular plants in one group. Lygophyta and Pterophyta, one group. Ne? Seed plants, then we have gymnosperm and angiosperm. So they say 2, 3, 4. 2, 3, 4. Do you have 4? Yeah, non-vascular 1, seedless vascular plant 2, gymnosperm 3, angiosperm 4. Ne? 4 groups. Each of the following can be seen in some other plant groups of plant kingdom plant A. Heterosporous. Heterosporous is present. This is 1. Heterosporous is present in one group. Heterosporous is present here, 1. Heterosporous is present here, 2. And this is 3. Ne? So heterosporous. Heterosporous I'm taking as 3. Heterosporous present in 3 groups. Heterosporous in 3 groups. Then presence of xylem vessel elements. Xylem vessel elements. Gymnosperm seeds are not enclosed. Angiosperm seeds are enclosed in chambers. Xylem vessel elements. One group kilagannadu mamma. Xylem vessel element, one group. But if you take people gymnosperm, then it's going to be 2. Shall we take it as 2 then? Gymnosperm 2 kilogram kuneda. Xylem. This is A. This is B. C. Dominant gametophyte. C. Dominant gametophyte. Gametophyte is dominant only in 1. Gametophyte is dominant in 1. Right? And then need of external water for fertilization. That's D, ne? External water. External water for fertilization. External water for fertilization. This one need, ne? This one also need. Conifers and cycles need. External water for fertilization. I get 3 once again. Need external water for fertilization. I get 3. I get 3. External water for fertilization, how many? 3, ne? This one, Pogonatum, Lycophyta, Pterophyta needed. Zygodophyta needed. Not external water, ne? External water, they say. Need external water for fertilization. Need external water for fertilization. Need external water for fertilization. Need of external water. External water, then I get 2 external water. Non-vascular plants, this one. Ceacus need internal water. Angiosperm also does not require any water. So external water, I get 2. I get 2. So if you take order CB, 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 xylem. Xylem, how many people? Now, what is xylem? Xylem vessel elements, into xylem vessel elements, these 2, ne? CB, CB, A, CBA. I think D comes here. CB, CB, DA. I don't have anything with 4. Yeah, we don't have anything with 4. Which of the above features arrange only 1, 2, 3, and 4 groups in correct sequence? Need of external water for fertilization. Each of the following features can be seen in some of the groups of kingdom Plantae. Each of the above features are arranged, seen only in 1, 2, 3, and 4 groups. 4 groups D. Need of external water for fertilization. I don't have a proper answer now. CB, CB. I have given the answer as 4, ne? CBAD, CBAD, but little complicated, ne? So we'll leave that question, people. Question number 8. We'll leave this question 8, right? Question doubtful. Huh? So it can be either these 2, 3 or 4. CABD can't be, ne? Right? Either third or second. I don't have a proper answer for that. Moving question number 9. Question number 9. Which of the following statement regarding vascular tissues of plant is correct? Which of the following statement regarding vascular tissues of plant is correct? Vascular tissues of plant is correct. Phloem of gametophytes. Oops. Right. Question number 9. Question number 9. Which of the following statement regarding vascular tissues of the plant is correct? Phloem of gametophyte has sieve tube elements. Phloem of gametophytes have sieve tube elements. We can't take that neither. Gametophytes don't have sieve tube elements. That is wrong. We'll take netophyte. Which of the following statement regarding vascular tissues of plant is correct? Netophyte. Sorry, I saw it as gametophyte. Phloem of netophyta has sieve tube elements. Netophyta sieve tube element wrong. Xylem vessel elements, not sieve tube elements. Wrong. Sieve cells of bryophyta transport organic food. Sieve cells of bryophyta transport organic food. Now, sieve cells are present in vascular plants. Bryophyte is not a vascular plant. You can't take that. Wrong. And then people, sieve tube element have pits between themselves. Sieve tube elements have pits between themselves. Sieve tube elements are having pits. Wrong. They have actually type of modified plasmodesmata. One sieve tube element to another sieve tube element, they are plasmodesmata. Wrong. And then seedless vascular plants do not possess sieve tube elements. Seedless vascular plants do not possess sieve tube elements. That is correct. Seedless vascular plants do not possess sieve tube elements. Correct. Sieve tube elements are present in Angiosperms. C-tube elements are present in angiosperms. Seedler vascular plants do not possess a Hari statement. Companion cells are found in Zygodophyta. Companion cells are present in angiosperms, not in Zygodophyta. Wrong statement. So we got the answer for 9th question. Fourth one is the answer. Fourth one is the answer. Question number 10. Question 10. Following are characteristic features of fungal reproduction. Characteristic features of fungal reproduction. Fungi. We learn fungi. Ne? So let's find the relationship. APX, Chytridiomycota P. Chytridiomycota zygosporangium wrong. Chytridiomycota, they don't have zygosporangium. Motile zoospores are there. You can't take that wrong. Right? Chytridiomycota wrong. And then people DSX, DSX. Check that DSX. Basidiomycota conidium. Wrong neither. Basidiomycota can't be conidia. That is ascomycota. Conidia, asexual reproduction. Ascomycota. Wrong. Right? You can't take that. It is wrong. And then people BPX. BPX. Let's check BPX. BPX. Let me erase these previous lines. BPX. How about that? B, P, and X. BPX. Zygomycota, zygosporangium, and sexual reproduction. Correct, Ned? Perfect. That's the answer. Ned. CQY. 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 How about that? C, ascomycota. Q, ascus. Y, asexual reproduction. Ascomycota, ascus, asexual is wrong. Why it is wrong, people? Because that is sexual reproduction. Eh? That is sexual reproduction. Wrong statement. And then the last one. So this is also wrong. CQY. CRY. CRY. Check that. CRY. CR and Y. Ascomycota basidium wrong. Ne? Ascomycota basidium also wrong. Ascomycota basidium wrong. This one also wrong. So we got the answer now. Answer is 3. Answer is 3. How many of you got that correct? How many of you got this one correct? Please raise your hand up. How many of you got this correct, people? Yeah, I have Vinuk got it correct. Anyone else? Oh my God. Oh, so many people. You all raise your hand. Huh? 9 people raised their hand. So I'm glad. I'm so happy. Eh? Not tired at all. Question 11. Lamai. Question 11. So I have some bright students in the class. Good. You do this. You're going to improve. Definitely. No doubt. Right? Support this night time work. Busy day. I know that you are like from the morning you are busy learning. And then traveling, and then in the night, again, once again, paper discussion. But for a short time period, maybe not even another year, right? You don't have another year for the examination. So try this short time period, work hard, right? That will take you to the success, right? So don't worry. Right. Question number 11. Question number 11. Select the correct statement about kingdom protista. Select correct statement regarding kingdom protista. Protista, correct statement I ask. Most of them are colonial and multicellular species. Correct statement regarding kingdom protista. Colonial and multicellular. What do you say about that, people? Protistants, colonial and multicellular. Most of them are colonial and multicellular species. There are unicellular ones available too. Multicellular, most of them are colonial and multicellular. Right? Protistans. They say most of them are unicellular, others are colonial or multicellular. That is what they say. So most of them are unicellular and colonial is a wrong statement. Right? We can't agree with that. Right? Most of them are unicellular and then others are colonial and multicellular. Sharp neither. These statements are sharp. Right? And then no symbionts. No symbionts. Wrong neither. Symbionts are present. Pellicle present in all unicellular forms. Pellicle present in all unicellular forms, wrong. Pellicle is available in Euglena pellicle present and Paramecium also pellicle present. So I don't agree with that. Wrong statement. Cell wall present in Gelidium, Sargassum and Ulva. Cell wall present in Gelidium, Sargassum and Ulva. That is correct. That is correct. Golden brown algae are multicellular. Golden brown algae are multicellular. Golden brown algae ke diatoms. So that is wrong. Fourth one is the answer for that question. Fourth one is the answer. Right? That's not correct. Golden brown algae, what do you call diatoms? Diatoms. Question number 12. The keratin found in the body, B, internal fertilization. And then C, people glands found in the body. And D, presence of teeth and aquatic habitat. Which of the above are common to reptiles? Keratin present, Keratin present, that is correct. And then internal fertilization, correct. Present, B also correct. So A correct, B also correct. Glands found in the body, wrong. Not correct. Presence of teeth, reptiles have teeth, aquatic, no, teeth absent in aves, birds. Aquatic habitat also we can which are the above common to reptiles? Wait. I have taken the question wrong. Wait, I think I need to make a small correction. That's not the question. What they ask, what is common to reptiles, aves and mammals? What is common to reptiles, aves and mammals? Right? Keratin found in the body. Keratin. Aves you can take, mammals you can take, and then reptiles also you can take. Correct. Then second one, internal fertilization. Reptiles internal, aves internal, and then mammals also we can take as internal. Correct. Glands found in the skin, you can't take that. Process of teeth, you can't take that. Aquatic habitat, you can take for all three. Aquatic habitat. So A, B, E. A, B, E, second one is the answer. Second is the answer. A, B, E. Question number 13. Question number 13. Select the correct statement regarding primary structure of the root of monocotyledon plants. Primary structure of the root of monocot plants. And moving to next page. Monocot plant root. Right, monocot plant root. Primary structure. Multicellular root has produced by some epidermal cells. Question is the correct statement regarding primary structure of the root of monocot. Primary structure. Multicellular root hairs, wrong neither. They have unicellular root hairs, wrong statement. Collenchyma cells are mostly present interior to the epidermis. Collenchyma present, mostly present interior. No collenchyma in the root. Collenchyma absent in roots. Outermost layers of the cortex. Outermost layers of the cortex is endodermis. Outermost layer of the cortex is endodermis, wrong. Why it is wrong? Innermost layer is the endodermis, not the outermost layer. Wrong statement. And then pericycle involves in formation of lateral roots. Pericycle involves in formation of the lateral roots. Pericycle involves in formation of lateral roots. That is correct, neither. Pericycle, but the question is regarding primary structure. Select the correct statement regarding primary structure of the root. So it says pericycle involves in formation of lateral roots. That is correct, but primary structure is not a valid statement. Endodermis is a single cellular layer without any intercellular space, and that is correct. So we'll go with the best answer 5. Best answer is 5. Question number 14. Which of the following responses is correct regarding transport of xylem vessel and phloem C tubes? Which of the following is correct? Correct statement regarding. Oh. Right? Which of the following response is correct regarding transport in xylem vessel elements and phloem C tube elements? Xylem vessel element, passive transport. That's correct, neither. Passive. Phloem is active transport, correct. They are asking correct regarding transport of xylem vessel elements and phloem C tubes. Right? Phloem C tubes kila kiyanne. Enam podi prasnayak kiyanawa. Not in phloem ne. Phloem C tubes. C tubes. That means from one C tube element to the other C tube element. Therefore, we'll take that as wrong. Why it is wrong? You learn phloem loading is active. Phloem unloading is passive. Flowing from one C tube element to the next C tube element is a passive process. It happens along the pressure gradient, ne? Pressure flow. Here they say people, xylem vessels and the phloem C tube, phloem C tubes. Not in the phloem. Right? So in both we can take as passive processes. So this first statement is wrong. Occurs bidirectionally. Wrong, ne? Occurs unidirectional, wrong. It should be other way around. Xylem is unidirectional. Phloem is a bidirectional one. Transport of water and minerals only. That is correct. Transport organic compounds. Wrong, neither. Transport organic compounds. Transport water and minerals. Organic compounds only. Some minerals can go through. Right? Some minerals can transport. What you learned, people? What you learned? Did you learn potassium available in the phloem sap? We mentioned maybe potassium available in the phloem sap. Can you remember that? Somewhere I can remember potassium in the transport materials, 30% by weight. Phloem sap aqueous solution that flows through C tube differ from xylem sap mainly because contain sucrose, may also contain amino acid, hormones, and minerals. Minerals amino acid, hormones, and minerals. Right? And so I think that's enough, ne? Minerals are present. So this one, organic compounds only wrong. Why? Minerals also. And then transport occurs in the apoplast pathway. That is correct. Transport occurs in the symplast, correct. And then transport occurs under hydrostatic pressure. Transport occurs under pulling force. Wrong, neither. Pulling force, no. Hydrostatic pressure, ne? Should be the other way around, ne? So we'll take people fourth one as the answer. Fourth one as the answer. Question number 15. Question number 15. What did you mark for 15? Come on, Lamai. Question number 15. Correct statement regarding cholenchyma cells. Cholenchyma cells, correct statement. They are generally spherical. No, not spherical. right? You can't take that the collenchyma as a spherical cell type, right? Collenchyma generally spherical. We learned that collenchyma cells are somewhat elongated. So I'll take it as wrong, right? The cells provide only mechanical support. These cells provide only mechanical support. Um, Only mechanical support. How about that, people? Parenchyma, mechanical support. What the resource book says about it. It allows these cells to grow. I'll read the resource book. It says generally elongated. They are unevenly young stems, even function mechanical support without restraining the growth. Give mechanical support to leaves and stems. They provide only mechanical support without restraining growth. Functional maturity, they are live, flexible, elongated, and functional maturity, they are live, flexible, elongated. Right? So we'll leave that second statement. They found just below the epidermis in dicotyledonous stem. Just below the epidermis in dicot stem. That is okay, correct. And then they found in dicotyledonous root. No, not in the root, wrong. And then corners of the cells are thickened with suberin cellulose. So we have the answer now, third one, right? We have the answer, third one, right? So we'll take third as the answer there. Question number 16. Question number 16. Question number 16. When fully flaccid cell immersed in pure water, when fully flaccid cell is immersed in pure water, what do you call flaccid? Flaccid කියන්නේ shrunken neither. Plasmalized cell, right, people? Fully flaccid is plasmalized. When a fully flaccid cell is immersed in pure water, right? Flaccid cell is immersed in pure water. Exosmosis occur. No, neither. Pure water, water goes into the cell. So endosmosis occur. Psi P gradually decreases. Psi P gradually decreases. No. Psi P is going to increase. Wrong statement. At dynamic equilibrium, there is no net water movement. At dynamic equilibrium, there is no net water movement. Cell become fully flaccid at equilibrium. At dynamic equilibrium, there is no net water movement. A net water movement when a fully flaccid cell is immersed in pure water. At dynamic equilibrium, there is no net water movement. Dynamic equilibrium. No net water movement. Cell become fully flaccid at equilibrium. Fully flaccid neither. Cell is going to become turgid at the equilibrium. If it is pure water, it becomes fully turgid at the equilibrium, not fully flaccid. Protoplasts of the cells become swell and press the cell wall against the plasma membrane. That is correct. Right? I think fifth one is a better answer. At dynamic equilibrium, there is no net water movement. Dynamic equilibrium, there is no net water movement. When the fully flaccid cell is immersed in pure water, it comes to an equilibrium. Right? And that at the equilibrium, no net water movement. Water does not move in or out. It comes to some sort of a balance stage. Right? At dynamic equilibrium, there is no net water movement. Dynamic equilibrium. Dynamic equilibrium. This word dynamic is the problem. So let me check the note a little bit, people. Right. It says then a dynamic equilibrium is formed and there is no further net water movement if the cell has the maximum value for psi p. Therefore, water potential becomes zero. This matches the water potential of extracellular environment. Then a dynamic equilibrium is formed. then a dynamic equilibrium is on. At dynamic equilibrium, there is no net water movement. There is no net water movement. That is also correct statement, right? So I'll take two answers here. So I'm taking 3 and 5 as the answer. 3 and 5. Dynamic equilibrium, no net water movement, right? I'll take that also as an answer. So you can, uh, any arguments, people? Any arguments? No arguments, right? Right. Moving people to question number 17. Question number 70. Tradescantia epidermal peels were placed. Tradescantia epidermal peels were placed in sucrose solution with different concentrations. Epidermal peels were placed in. I'll take people, if you want, I can take water potential discussion next week. What do you say? Huh? Shall we do a full water potential discussion? If you like, we can do that. What do you say about that? Huh? Because again, I feel like I feel like water potential is, but then you have to vote for that. Let's see how many of you are going to vote for water potential. Only water potential, nothing else. Huh? Only water potential, nothing else. How many of you? Please raise your hand up. There are 36 participants. Let's see how many of you are going to raise your hand up for water potential. Like quickly, don't take much time. So I can decide. Only 4 people. Only 4. Quickly before it disappears. 9, 10, 11, 12. At least I need more than half. Ne? At least I need more than half. 16 only. Ne? 16 only. Still not half. Ne? 17. 17. Come on. Come on. Click it. 18. 18 is 36. One more. Right. One more. 19. Ne? So we can take water potential discussion. I think that's better. Ne? We'll have a short discussion on water potential. Calculations. Ne? We'll do that. We'll do it. We'll do it. Right. Okay. Put your hands down. Right. Water potential. See. Yeah. But thing is now you have forgotten. That's the nature, Lamai. That's the nature. You forget you forget. That's nature. So as I said today, ne, only thing is we have to now find some time and revise it, not getting upset about forgetting the things. That's the nature of the brain. Right. Question number 17, my dear. Today is too slow, ne? Question number 17. Tradescantia epidermal peels were placed in sucrose solution with different concentrations. Tradescantia epidermal peels were placed in sucrose solution with different concentration. Select correct statement regarding this experiment. Select correct statement regarding this experiment. Turgid, flaccid, and incipient plasmolysis cells can be observed. Okay, let's come to the board. Now let's come to this board. Check this one, people. It says Tradescantia epidermal peels. Royo, ne? Tradescantia epidermal peels. Tradescantia epidermal peels were placed in sucrose solution with different concentrations. Select the statement regarding this experiment. Different sucrose solutions. These are 2 different sucrose solutions. Different concentration. Select the correct statement regarding this experiment. Turgid, flaccid, and incipient plasmolysis cells can be observed under the microscope. When you immerse Tradescantia royo epidermal peels in different concentrated sucrose solutions, turgid, flaccid, and incipient plasmolysis. What do you say? Incipient plasmolysis is not a stage that you can see under the microscope. Incipient plasmolysis is not a stage for you to see under the microscope. There, you can see turgid cells, you can see flaccid cells, but you can't see incipient plasmolysis stage. So first statement is wrong. First statement wrong. So I say that first statement wrong. When sucrose concentration increase, more cells become turgid. Sucrose concentration increase, more cells become turgid. Wrong. Why, people? Cells become plasmolized. Right? Cells become plasmolized. Then third one, solution potential. Solute potential of the tissue is calculated based on the solution that would give 50% plasmolysis. That is correct. Cells of epidermis are observed under high power microscope. Cells of epidermis are observed under high power of the microscope. No, actually we use medium power. We use medium power, so you can calculate count. Now high power shows you only one cell, people. High power shows you one cell or maybe two. That's not a good count. And then people, if you use a low power, you can see hundreds of cells. That is also not good. Medium power is better. Do you understand that? There is a linear relationship between concentration of solutions and percentage plasmolysis. Linear relationship. Wrong, neither. You remember the graph? How about the graph, people? Graph is a sigmoid one. Graph is sigmoid. That sort of a graph. So how do you say it's a linear relationship? It is not linear. So I'm taking that also as wrong. So got the answer. Third one. Got the answer, third one. How many of you got this correct? Is there anyone in the class to get this one correct? Huh? Anyone in the class to get this correct? Third one as the answer. Anyone of you? None of you? Milu. Anyone? Umaria. Mushfira. All right. Some people, 5 people. Okay, that's good. Rufika. Rufaika. That's good. Right? I'm impressed with that. Right, coming to people, question number 80. Which element deficiency is not contribute in chlorosis in older leaves? Chlorosis in older leaves. Which of the following element deficiency is not contributed to chlorosis in older leaves? Not contributing. Did I give you a short note for that? Gave you a short note, but I didn't give you a poster yet. Right? Which of the following is not contributing for chlorosis in older? I can remember molybdenum and nickel. I remember molybdenum and nickel you can't take because it resolves older. It resolves older. Zinc. Cassie, Cassie, crinkle. Zinc. Zinc. Magnesium. Nitrogen. Fluorosis does not. I feel like nitrogen. Right? I feel like nitrogen. Then I'm wrong. It has to be zinc. No memory at all, people. I think the answer has to be zinc. Answer has to be zinc. I need to check that. Right? I also can't remember. I think zinc. The twinkle leaves reduce internode. Activate of minions. It says chlorosis between. I thought of young nitrogen. What is that? Old leaves, young leaves. Nitrogen, older leaves, particularly older leaves. No. So I'm wrong. So that is zinc. Second one. Second is the answer. That didn't work. Question number 19. Question number 19. Which of the following is correct statement? Which of the following is correct statement? Correct statement. Which of the following is correct statement? In some plants, seed development without fertilization known as parthenocarpy. Some plants, in some plants, seed developed without fertilization is parthenocarpy. Parthenocarpy, carpang kilagwe, carpy kilagwe, food ne, carpang, fruits. So therefore, parthenocarpy seed development without wrong. In some plants, fruit development from ovary without fertilization is known as parthenogenesis. Fruit development parthenogenesis again wrong. In parthenogenesis, egg is resulted by mitosis. Parthenogenesis. The parthenogenesis. Egg is resulted from mitosis and hence is haploid. Wrong, neither. Right? It is wrong. And then parthenogenesis, haploid ovum can be fused with. In parthenogenesis, haploid ovum can be fused with polar nucleus. That is correct. Parthenogenesis occurs naturally in banana. Parthenogenesis, the parthenocarpy. Parthenocarpy, people. That is parthenocarpy. So that is wrong. So we got the answer now. Fourth one is the answer. Fourth one is the answer. Fourth one is the answer. Right. Question number 20. Question number 20. Question number 20. Correct statement regarding question number 20. Takes long time to discuss 20 questions. It consumes almost 1 hour and 15 minutes. Correct statement regarding gravitropism. Correct statement regarding gravitropism. Statoliths are found in the cells of the root cortex. Statoliths are found in the cells of the root cortex. Statoliths are found in root cortex. Wrong name. Where statoliths are present? Where statolith present? Root cap neither. Root cap. Next one. Statolith cause lateral transport of auxin within the root. Ekahari. Cell elongation in root is initiated by high concentration of auxin. Cell elongation in root initiated by high concentration. Cell elongation in root is initiated by high concentration of auxin. Cell elongation in root initiated by high concentration of auxin. Auxin and root elongation. Auxin and root elongation. What they had mentioned about it, people? Auxin stimulates stem elongation in low concentration. Stem elongation in low concentration. Lateral. Promote formation of lateral and adventitious roots in auxin. So we'll take it as wrong, right? Wrong statement. Cell elongation in root is initiated by high auxin concentration. Cell elongation is root initiated by high auxin concentration. Gravitropism affects on lateral growth of the roots. Gravitropism affects. Gravitropism is affecting on lateral growth. Gravitropism affect on lateral growth of roots. Gravitropism affects on lateral growth of roots. Statolith aggregates at higher points. Statolith higher points of the root cap. Higher point of the root cap and redistributes calcium ions. Correct statement regarding gravitropism. Statolith aggregate at higher points. Statolith hypothesis. It says aggregation of statolith at lower points of the root capital. Again, to take a wrong name. So higher points, higher points, highest point wrong name. Low points. Gravitropism affects the lateral growth of the roots. Lateral distribution of auxin. Right? I'll read that for you. The calcium, as a result, calcium and auxin get accumulated at the lower side. At high concentration of auxin, cell elongation is inhibited, resulting slow growth. Higher concentration cell elongation is inhibited, right? Not promoted, right? So here, cell elongation of the root is initiated, maybe inhibited, right? And then auxin get accumulated in the lower side. At high concentration, auxin cell elongation is inhibited, and consequent root grows downwards. So gravitropism effect on lateral growth of roots. So we'll take that also as strong lateral transport of auxin within the root. Second is the answer. Second is the answer. Right, we'll quickly go with the other questions now, people. Quickly go with the other questions. Which of the chemical reaction take place in the bundle sheath cells of C4 plants? Which of the chemical reaction take place in the bundle sheath cells of C4 plants? PGA become G3P. Bundle sheath, it happens. REBP plus carbon dioxide 3PGA, it happens. PEP plus carbon dioxide oxaloacetate does not happen. It happen in the mesophyll cell. Malate to pyruvate conversion happen. Oxaloacetate to malate. Oxaloacetate to malate does not happen. So A, B, D. A, B, D must be the answer here. A, B, D is the answer. Question number 22. Question number 22. Select the molecule. Select the molecules produced during the photosynthetic pathways. Molecules produced during photosynthetic pathway. Produced during photosynthetic pathway. Right? Acetyl coenzyme A. Then acetaldehyde. Acetaldehyde. Acetaldehyde photosynthetic pathway. Wrong. Where acetaldehyde produced, people? Ethanol fermentation. Oxaloacetate, hurry. Pyruvate, we can take. NAD plus, wrong. So CD must be the answer. C and D must be the answer. Question number 23. Question number 23. Which of the following statement incorrect regarding elements? Crinkling of young leaves is deficiency symptom of potassium. Incorrect. Calcium, not potassium. Crinkle, little star. Calcium and zinc. Yellow and brown leaf margin will be deficient symptom of calcium. Yellow and brown leaf margin. What is that, people? Yellow brown leaf margin. I remember like nitrogen. I remember it like nitrogen. Yellow brown. Chop canes. Potassium. 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 Check that, people. Yellow brown leaf margin. Yellow brown leaf margin. Potassium, yellow brown leaf margin. Potassium. Right? Yellow brown leaf margin. Calcium, wrong there. Chlorosis. Chlorosis in young leaves is a deficient symptom of sulfur. Young leaves, sulfur. Death of meristems. Boron. Boron. Dead Mary's boot. It was a reduced internode. The length is deficient symptom of internodal distance. Deficient symptom of zinc. Internodal distance. Can't remember that. What is it, people? Boron there. Internodal distance. Can someone tell me what is internodal distance? Zinc glue. Zinc glue. Oh, the zinc is wrong. Reduce internodal length is deficient symptom of zinc. Zinc crinkle. Reduce internodal length. Reduce internode length is deficient symptom of zinc. Reduce internode length. Reduce internode length is deficient symptom of zinc. Reduce internode length is deficient symptom of zinc. Correct me? Correct me. So my given answer is wrong then. CDE. CDE must be the answer. CDE. Please correct that in the Marquez scheme. Which question is this? Question number 23. 23 C, D, E must be the answer. Question number 24. Which of the following statement is correct regarding enzymes? Correct regarding enzymes. Correct regarding enzymes. Biotin, one of the cofactor of the enzyme, is coenzyme. Biotin, one of the cofactor, is a coenzyme. Correct, neither? Correct. Substrate largely changes the shape of the active site. Substrate largely, wrong. And then enzymes are highly specific to substrate. Correct. Enzymes are considered as polymers. Yeah, correct. Because they are proteins. Some enzymes do not contain active sites. Some enzymes do not contain active sites. Wrong. So ACD. ACD 24. Correctly. Question number 25. Question number 25. Which of the following statement regarding transport of plant is correct? Regarding plant is correct. Plant loading is mostly a passive process. Wrong. Occurs either from sink to source to form occurs either from sinks to source or from source to sink. Which of the following regarding is correct? Occurs either from sink to source. No, neither. Source to no. Every time people source to sink direction. Sometimes the source and the sink can alternate, but every time source to sink direction. At sink, unloading of sugars occur passively. Correct. And then phloem translocation is explained by pressure flow hypothesis. Translocation explained by pressure flow. Hurry. Ascent of xylem sap occurs in both directions. No, one direction only. So we have CD as the answer there. CD is the answer. Right? CD is the answer. So that's all for the day, people. That's enough. We took almost the entire time period for this. Right? Anyway, thank you very much for joining with tonight's class. We'll complete the discussion of this one. Plus, I'll make a special program, people, for you to revise water potential. So those things we realize, people, once we do the questions only. You have to do the questions and understand your weaknesses. Give me a comment, right? I get a feedback when I check your papers. So give me a comment, your difficulties. So we'll try to fix them all. Slowly, we have time. So we'll work, right? So thank you very much for your participation, right? And then I'll meet you all soon, right? Okay, bye. See you. Good night.